Hello everyone. So I hope you all have followed my channel and you have learned with me cell structure part 1. If not, you can always follow the link given in my description to follow and subscribe my channel. In this class, I will show you cell structure part 2, where we would learn about some further basic details about the cells and their organelles and their inherent functions. So we will start this lecture with the basic differences between animal cells and plant cells. So at this point you already know that each eukaryotic cell has a plasma membrane, cytoplasm, nucleus, ribosome, mitochondria, peroxisome and vacuole. But there are some striking differences between animal and plant cells. Both animal and plant cells have microtubule organizing center. Animal cells also have centrioles attached, a complex we called centrosome. Animal cells each have a centrosome and lysosomes. However, most plant cells do not. Plant cells have a cell wall, chloroplast, and other specialized plastids, and a large central vacuole, whereas animal cells do not. So let us look at each one of these instances separately. First of all, a cell wall. Cell walls are present only in plants. It is a rigid covering that protects the cell, provides structural support and gives shape. Fungal and some protist cells also have cell wall. Prokaryotic cell wall's chief component is peptidoglycan. However, major organic molecule in the plant cell wall is cellulose, composed of beta-1,4-glucose units. Next is the centrosome and lysosome. These are present in animal cells. Centrosome is a microtubule organizing center found near the nuclei. It contains a pair of centrioles which each contain 9 triplets of microtubule. We will discuss about microtubules in the later part of this class. The centrosome replicates itself before a cell divides, and the centrioles appear to have some role in pulling the duplicated chromosome to opposite ends of the dividing cell. But its exact function in cell division is not clear yet, because cells that have had centrosome removed can still divide, and plant cells which lack centrosomes are still capable of cell division. So the exact function is not known. Animal cells have another set of organelles that most plant cells do not, lysosome. These are the cell's garbage disposal. In plant cells, the digestive processes takes place in vacuoles. Enzymes within the lysosomes aid in breaking down protein, polysaccharide, lipid, nucleic acids, and even worn-out organelles, which cannot function anymore. These enzymes are active at a much lower pH than cytoplasm. So the pH within lysosome is more acidic. Many reactions that take place in cytoplasm cannot occur at low pH. So again, the advantage of compartmentalizing the eukaryotic cell into organelle is apparent from here. Next we come on to the chloroplast, the most important organelle of plants, at least with respect of human beings' use. Like mitochondria, chloroplasts have their own DNA and ribosome, but chloroplasts have an entirely different function. All of us know that it carries out photosynthesis. Photosynthesis is a series of reactions that uses CO2, water and light energy to make glucose and oxygen. This is the process which makes the plants as autotrophs, which are able to make their own food, while animals are heterotrophs, who depend on other organisms for their food. Like mitochondria, chloroplasts also have outer and inner membranes, but within the space enclosed is a set of interconnected and stacked fluid-like membrane sacs called thylakoids, these structures. Each thylakoid stack is a granum. The chloroplasts contain a green pigment, chlorophyll, whose main function is to capture the light energy which then drives the photosynthesis reaction. Some protists also have chloroplast. Some bacteria also perform photosynthesis, but their chlorophyll is not relegated to an organelle. The central vacuole. Previously, we have mentioned that vacuoles are essential components. Plant cells each have a large central vacuole that occupies most of the cell's area. If you go to the first slide of this lecture, here you can see the major vacuole of the plant cell. The central vacuole plays a key role in regulating the cell's concentration of water. Suppose if you forget to water a plant for a few days, it wilts, right? That's because as the water concentration in soil becomes lower, water moves out of the central vacuoles. As the central vacuole shrinks, it leaves cell wall unsupported. This loss of support results in wilted appearance. The vacuole also supports cells' expansion. When the central vacuole holds more water, cells become larger without having to invest considerable energy in synthesizing new cytoplasm. Now we come on to the next set of organelles called the endomembrane system and proteins. This system is considered separately because this is involved with synthesizing proteins and their transport. So endoplasmic reticulum is the first part in the endomembrane system. It is a series of interconnected membranous sacs and tubules that collectively modifies proteins and synthesizes lipids, however, in two different places, rough ER and smooth ER. ER's membrane, which is a phospholipid bilayer embedded with proteins, is continuous with the nuclear envelope. Now, rough ER. It is called rough because of the ribosomes attached to a cytoplasmic surface, which gives it a studded appearance. Ribosomes transfer their newly synthesized protein into the lumen, where they undergo structural modifications like folding or acquiring some side chains. These modified proteins get incorporated into cellular membrane, the ER or ER's other organelle membranes. The proteins can also secrete from the cell. The RER also makes phospholipids for cellular membranes. If the phospholipids or modified proteins are not destined to stay in the RER, they will reach their destination via transport vesicle. That is, the vesicles we had described earlier, like they are buses in the cargoes or transport in our part 1 lecture. Now the smooth ER. The smooth ER is continuous with the RER but has few or no ribosomes on a cytoplasmic surface. Smooth ER functions include synthesis of carbohydrate, lipid and steroid hormone, detoxification of poison and storing calcium ion. In muscle cell, a specialized smooth ER, the sarcoplasmic reticulum, is responsible for storing calcium ion. These ions are needed to trigger the muscle cell's coordinated interactions and movements. The next part in the endomembrane system is the Golgi apparatus. So the vesicles can burst from ER and transport their content elsewhere. But where do the vesicles go? Before reaching their final destination, lipid or proteins within the transport vesicles still need some sorting, packaging and tagging. It's like the airport, so that they end up in the right places. Sorting, tagging, packaging and distributing takes place in the Golgi. We call the Golgi apparatus cis phase. The opposite side is the trans phase. Transport vesicles that formed from the ER travel to the cis phase, fuse with it, and empty their content into Golgi apparatus lumen. As the proteins and lipids travel through Golgi, they undergo further modifications. Most frequently, short sugar molecules are added. These newly modified proteins then tag with phosphate group or other small molecules to travel to their proper destination. Finally, these are packaged into secretory vesicles that burst from the Golgi's other phase, that is the trans phase. Some of these vesicles deposit into other cell parts where they will be used. Others fuse with plasma membrane and release their content outside cell. So this is a picture. This is the nucleus. This is the rough ER membrane. These are the ribosomes. This is the cis phase of the Golgi. The vesicles are carrying it to the Golgi. Here all the sorting, packaging, tagging taking place. Again the vesicles are budding from the trans phase with the proteins inside them, and they are being released outside the cell. The next part is the cytoskeleton. We have already described in the previous chapter or in the previous lecture that the cytosol is jelly-like or semi-solid-like due to the cytoskeleton proteins. So if you were to remove all the organelles from a cell, would the plasma membrane and cytoplasm be the only components? No. There would be ions, organic molecules, and a network of protein fiber that help maintain cell shape, secure organelles in specific position, and allow this also to move. Collectively, these are called cytoskeleton. Microfilaments, microtubules, and intermediate filaments. So, of the three types of protein fibers, microfilaments are the narrowest. They function in cellular movement and are comprised of actin molecules. ATP, the energy coin, powers actin to assemble its filamentous form, which serves as a track for the movement of motor protein called myosin. This enables actin to engage in cellular events requiring motion, such as cell division and cytoplasmic streaming. Actin and myosin are plentiful in muscle cells. When your actin and myosin slide past each other, then only your muscles contract. They also provide some rigidity and shape to the cell. Several strands of fibrous proteins that are wound together comprise intermediate filaments. They have no role in cell movement. Their function is purely structural. They bear tension, maintaining the cell shape, and anchor nucleus and other organelles in place. But they are very diverse. One of the intermediate filaments you are familiar with is keratin, the fibrous protein that strengthens our hair, nails, and epidermis. The last cytoskeleton are the microtubules. Microtubules are small hollow tubes, polymerized dimers of alpha-tubulin and beta-tubulin. Two globular proteins comprise microtubules walls. These have a diameter of 25 nanometer and are the widest component. They help the cell resist compression and provide a track along which vesicles move through the cell, like when they are needed to transport the proteins and other cargoes. They also pull replicated chromosome to opposite ends of a dividing cell. Like microfilaments, they also can disassemble and reform quickly. They are also the structural element of flagella, cilia, and centrioles. Now, extracellular matrix of animal cells. Cells in most multicellular organisms release material into extracellular space. The primary component of these materials are proteins, and the most abundant protein is collagen. Cells have protein receptors on their plasma membrane's extracellular surface. When a molecule within a matrix binds to the receptor, it changes the receptor's molecular structure. The receptor in turn changes the microfilament's conformation, and these conformational changes induce chemical signals that reach the nucleus and turn on and off the genes. Don't worry, we'll look into all of this in further details. For now, just look at the example how extracellular matrix plays a role. So blood clotting provides an example in cell communication. When the cells lining a blood vessel are damaged, they display a protein receptor, which we call tissue factor. When tissue factor binds with another factor in the extracellular matrix, it causes platelets to adhere to the damaged blood vessel's wall, and this stimulates the adjacent smooth muscle cells to contract and initiate a series of steps that stimulate the platelet to produce clotting factor, and thus the blood clots. So extracellular matrix is a space through which cells express some signals, express some response, and also interact with other tissues. There are another methods or another components through which cells connect with each other. These are the intercellular junction. For example, plasmodesmata. These are present in the plants. So long stretches of plasma membrane of neighboring plant cells cannot touch one another because the cell wall surrounds itself. How then can a plant transfer water and other nutrients from its roots to its stem and to its leaves? Such transport uses the vascular system primarily, but there also exist structural modifications which we call plasmodesmata. These are numerous channels that pass between adjacent plant cell wall, connect their cytoplasm, and enable transport of materials from cell to cell and throughout the plant, like this. This is the path through the cytoplasm. This is the junction which is called plasmodesmata. A tight junction is another subjunction. This is the watertight seal between two adjacent animal cells. Proteins such as claudin and occludin tightly hold the cells against each other. This tight adherence prevents materials from leaking between the cells. Tight junctions are typically found in epithelial tissues. The tight junctions of epithelial cells lining your urinary bladder prevent urine from leaking out into the extracellular space. So you can imagine how important they are. Last are the gap junctions. Gap junctions in animal cells are like plasmodesmata, in that they are channels between adjacent cells that allow for transporting iron, nutrients, and other substances. So, tight junctions don't allow the movement in spaces where they are not needed. Gap junctions allow the movement. Gap junctions develop when a set of 6 proteins in the plasma membrane arrange themselves in an elongated donut-like configuration, a connection like this. When the connection forces adjacent animal cells align, a channel between the two cells forms. Like this is a door, this is a door. They are aligning, so a channel is formed. So the molecules can move between here to here. Gap junctions are particularly important in cardiac muscle. The electrical signal for the muscle to contract passes efficiently through gap junction, allowing the heart muscle cells to contract in tandem. So this was the basic concept about cell structure part 2. That was all for today's class, but soon we will come up with revision questions, critical thinking questions, and MCQs on this chapter. Also, in the next lecture, we would move on to elaborate details about the cells such as its respiration, photosynthesis, plasma membrane, etc. So stay safe and stay tuned to the channel. Thank you.